Dear students, today I am going to discuss about the topics lymphoproliferation assay, mixed lymphocyte reaction, then transgenic mice and knockout mice. First, I will start with lymphoproliferation assay. Lymphoproliferation assay or lymphocyte proliferation assay is used to determine the lymphocyte activation and the cell-mediated immune responses. When the B lymphocytes encounter their specific antigens with the help of T lymphocytes, B lymphocytes are stimulated to undergo proliferation. And when the T lymphocytes are activated by the antigen-presenting cells and the cytokines, the T cells undergo proliferation. So the proliferation of both the B lymphocytes and T lymphocytes leads to clonal expansion and the initiation of the specific immune responses. So cells that are undergoing proliferation increase their rate of protein and DNA synthesis. The increase in DNA synthesis can be measured by adding a radio-level thymidine. So, lymphocyte proliferation assay actually measures the functional capability of lymphocytes to respond to antigenic or mitogenic stimulations. And the assay is a more direct test for immunocompetence than a total lymphocyte count. So these assays are the most frequently used in vitro tests to assess the cell-mediated response to a nutrition intervention. And mostly the investigators report a decrease in the response to a mitogen in both children or in adults with protein energy malnutrition. Decreases in cell-mediated responses occur early in the development of protein energy malnutrition, so that the lymphocyte proliferation is said to represent a sensitive measure of nutritional status. So here in this picture you will see that the B lymphocytes, here is your B lymphocytes and this is the T lymphocytes. Both the helper T cell and the cytotoxic T cell, means CD4 plus T cell and CD8 T cells. So when both the lymphocytes, means B cells as well as the T cells, they are mixed with a radio level thymidine, that is a tritium tagged thymidine, a radioisotope level DNA precursor. So thymine is a precursor, is a nucleotide. So it acts as a nucleotide analog. Thymidine means it's a nucleotide. So when these radioisotope leveled DNA precursor is added to the cell culture where both types of lymphocytes are present, the amount of tritium taken up by the dividing cells is correlated to the level of cellular proliferation. Here, cells undergoing proliferation are also metabolically active and increase their cellular level of dehydrogenase. So dehydrogenase and their product, that is NADH, nicotinamide and reduced nicotinamide adenine dinucleotide, and reduced nicotinamide adenine dinucleotide phosphate NADPH. So to track the levels of NADH and NADPH that is present into the culture of the proliferated or the growing cells, we can measure these two enzymes, or the enzymes metabolically active here, that is dehydrogenase, which is functioning here on NADP and NADH, and we can track, we can measure the level of dehydrogenase present at the means level, means at the cellular level of dehydrogenase present there in the culture, by using the test which is known as MTT assay that we have already discussed. So a tetrazolium dye we have used, which is normally yellow in color, but when interact with this enzyme, so they will form an insoluble purple formagen, and that insoluble purple formagen is the indicator of how much amount of cells they are metabolically active, and means what are the living cells and what are the dead cells, because the dead cells are not metabolically active. So dehydrogenase is is not elevated there. So the resulting purple color, the resulting purple insoluble product can be solubilized by using GMSO and can be quantified via colorimetric assay, say MTT assay. So MTT and tritium treated thymidine incorporation are the two common methods that are used to measure proliferation of lymphocytes. Here we can cytokines such as interleukin 2 can also stimulate T cell to proliferate. And the T cells proliferate in an IL-2 concentration dependent manner. So the level of the T cell proliferation can be used as a measurement of IL-2 concentration. And using IL-2 with a known concentration of as a standard, the concentration of IL-2 in an unknown sample can also be measured by its ability to stimulate T cell proliferation. So we can measure it. So the procedure is like that. Uh, uh, Here we have to use the cells, and first we have to some precautions should be taken when we are performing this lymphocyte proliferation assay. As because that we cannot open the plate or the tube containing the cells as well as the medium and the boxes that contains the pipette. It should be sterilized. It should contain sterilized tips, and we cannot open anything in open air. Everything should be open under the laminar airflow, and we have to wipe the surface of the laminar flow hood, laminar flow platform, and pipette man, our gloves should be properly wiped out using 70%. And when we are carrying out the experiment under the hood, we have to take precautions that all the mediums, all the plates, everything, all the utensils should be decontaminated first using 70%. And during the exercise, we cannot, means do not let your cells to come in contact with anything that is not sterile, including your finger and the tips that you are using for transferring media or the spent media cannot be touched at the surface of the wood and cannot be taken out from the laminar workstation, as well as you cannot touch outside of the medium bottom. So this particular type of proliferation assay is used as an in vitro substitute, similar to the in vivo delayed type hypersensitivity reaction. So to assess the overall quality and the character of the cellular arm of the immune response, we can use this. This proliferation assay is used to assess congenital immune deficiencies, transient immune compromised states, and more recently, the progressive immune deficiency in case of the HIV patients or AIDS patients, HIV infection. We can use this test. Next test I'm going to discuss about the mixed lymphocyte reaction. So this particular test is useful and can be considered as a model that should be used uh, by the pharmaceutical as well as the biotech industry to study alloresponsiveness to histocompatibility antigens. So in short, it mixes populations of lymphocytes together and measures the reaction that occurs using the mixed lymphocyte reaction. We can measure the disparity in major histocompatibility complex antigen between individual to individual. Particularly the potential immune activity or immune suppressive effects uh, of the drugs can be determined using this assay. So here also we have to co-culture. Means two type of lymphocytes can be co-cultured from means one from we have to take T cells from one individual and T cells from another individual, the second individual. Then we have to mix it and we have to go for co-culture. So if the cells of one individual recognized others' MHC molecule as a foreign, they proliferate and divide. And the proliferative response is usually measured by the process which has just now I have discussed this is tritium level thymidine uptake assay. There are two variants of the test. Means two ways of the test that are coming from the two individuals. The cells of both individuals are free to proliferate. And if we are using a one-way assay, the cells of one individual are irradiated or treated with a mitogen, treated with say mitomycin C, so they can proliferate, but the others do not. The cells of only one of the individual can proliferate. So there are two type of assay, two type of variants of the test exists. So here we are going to discuss about one-way test. So here we have to isolate the lymphocyte, and from there means we can isolate PBMCs from two individuals. Then PBMCs or lymphocytes isolated from PBMCs from one individual will be irradiated or treated with mitomycin C, and the second individual will not. So the irradiated cell, here you can see, here, this is the irradiated cell, the yellow one, the blue one is not. So cells from two individuals, one is irradiated, another one is not irradiated. So the cells that are irradiated or treated with mitomycin C, that lymphocytes, they will be stimulated, because here irradiation by using some radiolabeled isotope or treatment of mitomycin C will actually stimulate the lymphocytes that that but that cannot proliferate. So they are acting here as a stimulator cell. But there we are using this that can proliferate, but here they cannot proliferate. The cells from the two individuals are mixed, and after 3 to 7 days, both the T type of cells, means the T cells that have uptaken thymidine, means tritium level thymidine, and if the unirradiated lymphocytes contain alloreactive T cells, they will proliferate and differentiate to the effector cells as a result of recognition of foreign MHC molecule. So what is happening here? So here we have seen that the unirradiated cells, unirradiated lymphocytes, which contains an alloreactive T cells, means they will proliferate, not the irradiated cells, or not the mitomycin C treated lymphocytes. So we can differentiate this proliferative lymphocyte from the effector lymphocytes. Means that is actually stimulating but not proliferating. So these effector cells we can differentiate, we can sort out as a result of recognition of foreign MHC molecule. So here in this test you can see that these cells, the irradiated cells, are not proliferated, but the unirradiated cells, they are proliferated. So generation of cytotoxic T cells depends largely on differences in MHC class 1 alleles, means they are allogenic or allogenic MHC1. Now, this mixed lymphocyte test or reaction can act or can apply in different tests. The goal of this experiment was to check the effects of ciprofloxacin on intracellular expression of interferon gamma in monocytes and T cells during this particular reaction, multiple mixed lymphocyte reaction. Cells from two unrelated individuals were mixed and incubated under different conditions for 24 hours. All the reagents were added at the beginning of the test, and then we have to check the expression of interferon gamma via flow cytometry assay. So that we can check. Next, I'm coming to the transgenic mouse. So transgenic mouse is one whose chromosomes have been altered, so that altered deliberately, so that its genes contain foreign DNA. These genes are found in the nucleus of every cell of the body. So all of the mouse's cells contain the new DNA. The foreign DNA can come from any source, and maybe human, from another animal, or from another mouse. And the change in DNA usually means the cells gain a function, such as producing a new protein. For example, some transgenic mice contain proteins recognized by human immune cells. And these can be used to model the particular aspects of disease. The foreign DNA can sometimes mean a loss rather than a gain in function, as the new DNA might interfere with the biochemical pathway or prevent production of a particular protein. Transgenic mice are usually used as a model for understanding how gene regulate processes in the body. Because the effect of changing a particular gene can be seen on the whole organism. They are also used to study human diseases which are caused by errors in the way that the body produces certain proteins. For example, in hemophilia A, the crucial gene codes for a protein known as factor 8, which is needed for blood clotting. So to get some foreign gene sequence into every cells of the mouse, it is necessary to introduce the DNA into cell of very early mouse embryo that will contribute to the germline, means the cells that produce eggs or sperm. Now, where are these animals being produced? So the two common reasons are that transgenic animals are very useful as a disease model and produces or producers of substances for human growth. So they will express some gene, and the expression of these genes can be tracked outside the human. So some transgenic animals are produced for specific economic traits. For example, transgenic cattle are created to produce milk containing particular human proteins, which may help in the treatment of human emphysema. Other transgenic animals are produced as disease models. For example, genetically engineered mouse called oncomouse carrying a gene that promotes the development of various human cancers. So there are different types of. So this particular process, this is the process how the transgenic mice is prepared. So the two main techniques for introducing the foreign DNA into the mouse are either through pronuclear injection or use of embryonic stage. In pronuclear injection, the foreign DNA is injected into the pronucleus of a mouse egg, which forms just after it has been fertilized. The foreign DNA integrates into the genome at a random position, usually after the first one or two cell divisions have occurred. This means that the mouse will not carry transgenic DNA in all of its cells, and so will only be particularly transgenic. The transgenic eggs or sperms from these mice are then used to create the next generation of fully transgenic mice. When DNA is introduced into embryonic stem cells, it usually integrates randomly into genome, but if it has a similar structure to an existing part of the genome, it can be recognized by the DNA so that it undergoes homologous recombination. And a single copy becomes integrated into the genome at a specific location. So these embryonic cells then need to grow and are injected into a host embryo, becoming part of the mouse which grows from that embryo. The mouse grown from the host embryo is known as chimera and is formed from the embryonic cells of two different mice. Some of the sperm produced by the chimera will be transgenic containing the foreign DNA, and when this sperm fertilizes a normal egg, the mouse which grows from it will be fully transgenic with foreign DNA in every cells. So there are some examples of transgenic mice. So the examples are oncomice, supermice, doggy mice, cancer-resistant mice, mighty mice, such and such. So this mice has been used for the different purpose, mostly mostly for the synthesis of some protein, for checking the oncogene or the expression of oncogene, like that. Now I'm coming to knockout mice. The more recent development of knockout or knocking strains of mice during 1980s was a major advancement in genetics. This technology allows particular gene on the DNA strand to be altered, usually removed, but they may also be inactivated or inserted. This allows researchers to determine the exact function of a particular gene. And these genetically modified mice have provided excellent models of many human diseases, which could not be studied in animal before. The sequence and analysis of the mouse genome has allowed many genes to be targeted and studied using this particular technology. So the creator of the first knockout mice were awarded the 2007 Nobel Prize in medicine. So a knockout mice is actually a genetically engineered mice, and this particular technique, which led to the creation of knockout mice, was developed in bacteria by Joshua Lederberg, for which he has got Nobel Prize in 1958. So it's a genetically engineered mouse in which researchers have inactivated or knocked out an existing gene by replacing it or disrupting it with an artificial piece of DNA. The loss of gene activity often causes change in a mouse's phenotype, which include appearance, behavior, and other observable physical as well as biological characteristics. So this is the model how you can knock out a gene. So this is the cell resistant to both G41B and uh, gens and Gansi and ganciclovir, then and then and antiviral drug. So this is the original allele. A is the original allele, and A star is the replacement. Now we are targeting this gene, we are replacing this gene with a new DNA sequence. So knockout mice are important animal as an animal model for studying the role of genes which have been sequenced but whose functions have not been explored. By causing a specific gene to be inactive in the mouse and observing any differences from the normal behavior or physiology or biochemical uh, properties, researchers can infer its probable function. So, to identify its probable function, here here the scheme is showing two genes. A is the original gene, A star is the replacement gene, and this is neo-R, that is antibiotic resistance, neomycin antibiotic resistance gene, and it's relative like the drug G418, which is lethal to mammalian cell. And one more thing, TK is a gene that encodes thymidine kinase, an enzyme that phosphorylates the nucleotide analog ganciclovir. So DNA polymerase fails to discriminate against the resulting nucleotide and insert the non-functional nucleotide into freshly replicating DNA. So ganciclovir kills cells that contain the TK gene. So we can understand the function of a particular gene by using this knockout model mice. So this knockout mice can be synthesized by interbreeding, and by producing or gene targeting. And this technique allows a specific gene on the mouse genome to be altered by replacing it with a similar genetic sequence which has been modified to certain mutation. So this modified gene contains a mutation. The mutation often prevents the gene from functioning. And when genes are knocked in, a mouse gene is often replaced by a similar gene from the human genome. The mutant gene is created in a bacterial plasmid, which is injected into mouse embryonic stem cells, usually from male mouse. These cells are formed a very early mouse embryo and will divide to form every type of cell in the body. So we can synthesize this mouse. So first, these cells, they are not a complete mutant, a complete knockout. The stem cells are then implanted into the host embryo to grow. Mice with different colored fur are usually selected as the host so that it is clear which mice have the mutant genes. When the mouse is born, it will be chimera. As only here you can see it will be a chimera. So as only some of its cells will be modified, and this mouse will have fur of both colors. When sperm from the chimeric mouse fertilize a normal egg, some of its offspring will carry a single copy of that mutant gene. These mice will have same colored fur as the mouse whose DNA was altered originally. So here you can see this is the normal mouse and this is the altered mouse, means gene knockout, so gene targeted mouse. So there are some examples. We can use this for the treatment of different diseases, human diseases, genetic disorders, and to identify function of a gene, and if the gene is misbehaving, mutated, then how it is malfunctioning. So in the study of some diseases like cystic fibrosis, thalassemia, and other different type of cancer, we can use this knockout mice. And such models are very useful because they can be used to investigate the biochemical and physiological aspect of the disease and for the development and testing of drugs. Now, these are some examples of knockout mice. And there are many examples of knockout mice. Cat mice, carboxypeptide deficiency. Uh, they're prone to obese due to carboxypeptidase E deficiency. And strong mice with a disabled uh, myostatin gene, cool tolerant mice, etc., that are lacking sodium channel, which causes pain when exposed to cold. So, these transgenic mice or genetically modified mice are used extensively in research as models of human disease. They have been used to study and model in obesity, heart disease, diabetes, arthritis, substance abuse, anxiety, aging, Parkinson's disease, like that, and also in toxicological study. In drug development, we can use this. whether the proposed drug is a toxicant to the normal cell, and if it contains toxicity, how much is the toxicity? What is the level of toxicity that we can check using this transgenic mice having modified, means these are genetically modified mice containing a human gene. That's all for today.